Hello everybody, my name is Alejandro Gonzalez and today I will talk about our project Phylogenetic Relationship of Mole Crabs of the genus Emerita derived from mitochondrial DNA in sandy beach of Mexico Atlantic coast. So mole crabs are abundant crustaceans that inhabit the intertidal zone of sandy beaches worldwide. However, their phylogeny is poorly understood because of the small number of studies that have been conducted. Due to this lack of phylogenetic analysis, the group remains dynamic, which means the reassignment of species from one genus to other within the same family is frequent. In the present study, we examined for the first time the phylogenetic diversity, relationship, and genetic variation of the genus Emerita in marine ecoregions of the Atlantic coast of Mexico using mitochondrial DNA. Fieldwork was carried out in 16 localities using a 500 micron sieve. The macrofauna was obtained included as specimens of the genus Emerita. Once sieved and in reactive alcohol, the organisms were taken to the laboratory for their taxonomy identification. Then, DNA extraction were performed with the Kiagen Fast DNA tissue kit. Then we amplified COI and 16S mitochondrial genes. Upon receiving the nucleotide sequences from Matrogen Inc., the chromatograms were checked to verify the quality of the sequences, and subsequently the alignments were performed manually first of all. Later, we corroborate with an automatic alignment using codon code software and Clustal X2 with a muscle algorithms. Finally, the molecular analysis we're performing using RStudio software with the Fangor package, where neighbor joining maximum likelihood and UPGM trees were created, looking for the trees that best adjust the sequences. Optimized parsimony parameters, nucleotide substitution models, the bedfist, and bootstrap analysis were obtained. So we run an optimized maximum likelihood tree of the GTR model with 10,000 bootstrap pseudo replications. In the Merita phylogenetic performance, out of 25 sequences were obtained 637 base pairs corresponding to mitochondrial DNA, in which we found 506 conserved characters, 129 variable characters, of which 92 were parsimonious. Using a cake information criterion number, we obtained that the best model was the general time reverse model or GTR model. So we can construct a maximum likelihood tree that had well supported topology. Our two species are well defined, supported by a rooted outgroup called Lepidopa. As well, the correlation between the phylogenetic tree and the marine ecoregions led us to determine a strong relationship between Emerita benedicti and the terrigenous sediment ecoregion. On the other hand, Emerita talpoida is more related to carbonate sediment, like in the Campeche-Yucatanian inner neritic ecoregion. However, a special revision needs to be performed because we find three Emerita talpoida out of the carbonate sediment ecoregion, as we see here in Nautla, Veracruz, and in Santana, Tabasco. As a conclusion, we have two species of Emerita in sandy beach in Mexico, Atlantic coast, that could be delimited by the sediment or the ecoregions, as we can see in the sand composition. Even when the relationship between clades are well represented, some incongruences was found. So we suggest to enlarge core region and add a nuclear region, so we can perform a more precise analysis. And finally, it would be important to incorporate more location, so perhaps we could strengthen the analysis and complete a full solved phylogeny. We want to thank to everyone who made this possible. Thank you very much.